Hi, my name is Vicky, and I have conducted a project about the effects of menorrhea on the persistence of semen in sexual offence victims in my third year project. So, just a little introduction. The effects of menorrhea, which is actually commonly known as period blood, on the seminal persistence is actually extremely under-researched, despite its very importance and a very often encountered in rape and sexual offence cases. For example, within the literature I found, Dakado et al. 2011 discussed that there is an extreme lack of research into the effects of menorrhea on the vaginal environment and the vaginal secretions, therefore making it impossible to comment or actually predict its effect on seminal persistence or on any kind of presumptive testing or confirmatory testing. Despite the lack of research into menorrhea as a variable, all presumptive and confirmatory testing are carried out as normal within today's practice, as if the variable wasn't actually there, therefore possibly jeopardizing the collection or the outcome of the final results. They're actually unknown and cannot be predicted due to the lack of research. These samples are very often collected by the early evidence kit in the police force, and then their further examination takes place at a SARC facility where collection by cotton swab takes place, and then they get sent to a forensic laboratory where the cotton swabs actually undergo presumptive and confirmatory testing. So let's talk a little about the menstruation versus the persistence of semen. So STAG 2007 gathered persistence data, which is based in a clinical forensic medicine book, and it shows that after vaginal intercourse, the semen can be detected in the vaginal environment for around 24 hours, are likely to be found 3 days later, and occasionally can be found up to 7 days after the initial intercourse. This is actually a very minor encounter. Stark further argues that the longer time for persistence are actually exceptions and not a rule, and are dependent on a variety of variables which have not been researched, such as the time or washing by the victim, any variable which can really affect the amount of semen still present. I also found within the literature that RUP 1969 stated that the menorrhea allows the sperm to retain motile for a longer amount of time within the endocervical region of the vagina, maybe due to the fact that it's a lot more fluid and it allows it to rest there for quite some time. However, Cardell et al. 2016 does not agree with RUP 1969, and he states that there is a lack of data into the effect of menorrhea on seminal persistence. Therefore, it cannot be concluded if it has an effect on the seminal persistence or if it just doesn't affect it at all. Rupp and Cardell actually do agree on a point, which is that there's extremely lack of research into the variables which affect the seminal persistence, and they also agree that the time has a very big effect on the seminal persistence, decreasing its amount and its quantity and quality in the overall investigation. My aim within this research was to determine if the menstrual cycle influences the persistence of semen in sexual offence victims. Therefore, my objectives were to determine if menorrhea present has a significant effect on the seminal persistence, to determine if the time influences the quantity and quality of spermatozoa present, to determine if the presence of menorrhea influences their test use, such as the presumptive and confirmatory testing, can it cause ineffective false negatives or false positives. And then my null hypothesis was that the menorrhea end time has no effect on the persistence of semen, and my alternative hypothesis was that the menorrhea end time influences the persistence of semen. So my preliminary research consisted of identifying the effective presumptive testing. So all the presumptive testing I've used, which was the PSA, acid phosphatase, ultraviolet light, were actually subjected to samples of only menorrhea and only neat semen. They were compared to each other to see if they're, for example, in acid phosphatase, if the colour changes are very similar, if menorrhea can cause false positive or negatives, will the test come up positive if there's only blood present, or will it only become positive when there is semen present? I also conducted controls and identifying the false positive and negatives, as I stated a second ago. I also established standard volume of Menorrhea and semen within the project. So the all amount of semen and Menorrhea I have collected from the participants, I had gathered an average from, and I used an average then in every single attempt of the experimental. I also had to establish if the drainage within the gynecological model will be too fast for me to leave the model for 20 hours. So that was tested, and the model was of the biological samples were deposited and the model was left, put in a vertical position, and I found that actually drainage occurred extremely fast. Like within the first 10 minutes, all the fluid came out and it wouldn't have been possible. So I had to tighten the vaginal canal of the gynecological model to try and retain as much fluid as possible. The preliminary experiments were necessary due to the fact that if I did not do these tests, my main experimental wouldn't have been successful. The biological fluids would have come out too fast. I wouldn't have been able to collect samples after 20 hours. I wouldn't have known my Menorrhea and semen samples, the amount I need to use, if the presumptive testing for false positive or negatives. I wouldn't know if my results have a significant meaning. Within my main experimental, I've used a gynecological model to simulate a female victim. The biological fluids were deposited within the gynecological model. So 4 milliliters of pure semen were deposited at one time, and then 4 milliliters of semen and 3 milliliters of Menorrhea were deposited a second time. The model was then lying down in a horizontal position for about 10 minutes, as due to the research I found by Salem et al. 2000 actually states that the bed rest improves retention of seminal fluid by 13.3%. Therefore, I decided to use that within my research to increase the retention time within the gynecological model. The model was then put in a vertical position where quite a big amount of seepage happened. The model was then left for 2 and 20 hours. The evidence collection after 2 and 20 hours took place using cotton swabs. The cotton swab was inserted into the gynecological model and then taken out. These swabs were then subjected to presumptive testing of acid phosphatase, PSA, and ultraviolet light, where positive reactions were indicated by purple colour change, fluorescence, and the appearance of red line in a control and test window. Further confirmatory testing was conducted using microscopy. The swabs were centrifuged, deposited onto the microscopical slide where H&E staining was applied. The H&E staining visualized the spermatozoa present within the microscopical slide. The slides were then observed, and the amount of spermatozoa was extrapolated from the assumed existing trend. And they were further then compared between the pure semen sample and the mixed bodily fluid sample to see if there is any difference. From the data gathered, I then conducted statistical tests to see if there is a statistical difference between the time and the bodily fluids between the both samples. Let's talk about the results and statistical tests. So ultraviolet light test showed a 100% positive reaction, which was represented by fluorescence after 2 and 20 hours, whereas in mixed bodily fluids, the positive reaction decreased to 60% after 2 hours and further decreased to 20% after 20 hours. Acid phosphatase showed a 100% positive reaction, which was represented by a purple colour change for both neat semen and mixed bodily fluids after the 2 hours, whereas after 20 hours, the neat semen decreased to 80% and mixed body fluids remained at 100% of positive reactions. The PSA showed a 100% positive reaction for neat semen after 2 hours. However, after 2 hours, the mixed bodily fluids reactivity decreased to 80%, whereas following the 20-hour mark, both bodily fluids showed 100% positive reactions when analysed. So the confirmatory testing of microscopy showed that there was an average amount of neat semen samples after 2 hours. An average amount of spermatozoa found was 141, whereas after 20 hours this amount decreased to 59 of visible spermatozoa. In mixed bodily fluid samples, the average amount of spermatozoa found after 2 hours was 125. However, after 20 hours, this number decreased to 47. All samples, however, showed a standard deviation, a very high standard deviation. Therefore, there was a wide disperse of data from the true mean, with a presence of quite a lot of outliers. So, from my data gathered from the microscopy and the extrapolated data from the assumed trend within the microscopical slide, I conducted a two-way repeated ANOVA test, which was a statistical test conducted in order to identify if the two independent variables in combination, which were the bodily fluids and time, have an effect on the independent variable, which was the persistence. The statistical analysis revealed that there is a statistical difference between the bodily fluids tested, so there is a difference between neat semen samples and mixed bodily fluid samples, and the persistence of spermatozoa between them differed when there was an addition of Menorrhea. The statistical analysis also showed that there was no statistical difference between the decrease of spermatozoa between the two time frames, so the 2 and 20 hours, between the both body fluids. That means that they decreased at a very similar rate, and the amount that they decreased by was extremely similar. Therefore, from my research conducted, I believe that this experiment had a beneficial impact on the failure collection of samples from sexual offence victims. As now the effect of Menorrhea has been researched a little bit, we know that it affects the persistence of neat semen and that it does decrease. It also makes the spermatozoa actually a lot harder to see during the microscopical analysis due to the H&E staining, as the H&E stains red blood cells, which are within the Menorrhea. The spermatozoa was a lot harder to visualize. Therefore, maybe a different staining technique should be utilized in the future research to see if the staining technique actually does have an impact. Well, that's the end of my presentation. Thank you very much for listening. Hope you do really well with your next year projects. Wish you good luck and I hope you enjoy it. Thank you.